Welcome. In this video we are going to discuss about chloroplast transformation. Now in case of plants, the genetic material is distributed in nucleus, chloroplast and mitochondria. Okay, there is genetic material in nucleus as well as in chloroplast and in mitochondria. Now each of these three compartments here have their own genome and expresses heritable traits. Okay, they expresses heritable traits and they have their own genome. Now chloroplast is present in photosynthetic eukaryotes, that is plants. Now there are about 300 chloroplasts in one plant cell. Now the genome of chloroplast is circular double-stranded DNA and it varies in length. Okay, its length varies from 120 kb to 190 kb. Okay, so there are 300 chloroplasts in one plant cell. The chloroplast genome is circular double-stranded DNA varying in length from 120 kb to 190 kb. Now in most species, chloroplast is maternally inherited, that is 80% of times, in case of angiosperm plant species. Okay, the chloroplast is maternally inherited. Now chloroplast genome is not influenced by polyploidy, gene duplication and recombination. Okay, these factors don't influence the chloroplast genome, and these features are widespread features in case of plant nuclear genome. Okay, but they don't influence the chloroplast genome. Okay, polyploidy, gene duplication and recombination. And that is the reason why the chloroplast DNA varies a little in size, structure and gene content in angiosperm, because it is not subjected to these features. Okay, that is the reason why it varies little in size, structure and gene content. Now chloroplast transformation involves the delivery of DNA into the chloroplast. Now during chloroplast transformation, the DNA has to be delivered through cell wall and three other membranes, one of which is plasma membrane and two chloroplast membrane. Okay, to deliver the DNA into the chloroplast, we have to cross cell wall and three membranes, including one plasma membrane and two chloroplast membrane. Now efficient chloroplast transformation have been achieved by both, that is particle bombardment method as well as polyethylene glycol mediated transformation. Okay, both of these methods are used for efficient chloroplast transformation, that is particle bombardment method and polyethylene glycol mediated transformation. Now this PEG mediated transformation of plastids, that is chloroplasts, involves enzymatically removing the cell wall and making a protoplast and then exposing that protoplast to the purified DNA in the presence of PEG. Okay, first you enzymatically remove the cell wall from the cell, then you get protoplast out of it, then you expose that protoplast to the purified DNA in the presence of PEG, and that is how then your DNA will be transferred in the chloroplast of this protoplast. Now protoplast first shrinks in the presence of PEG and then it lies due to disintegration of the cell membrane. Okay, in the presence of PEG, first it shrinks and then it gets lysed due to disintegration in the cell membrane. Now we remove PEG before the membrane is irreversibly damaged, and removing PEG reverses the process. That is, the cell that got shrinked will become normal again if you remove the PEG, and you remove it before the membrane is irreversibly damaged. Now the other strategy that we discussed is biolistic delivery, and this biolistic delivery for chloroplast transformation or any other cell transformation is a routine system in most of the laboratories. Now, flowering plants contain variety of plastids, including chloroplast, leucoplast and chromoplast. Okay, these all are the plastids which are present in the flowering plants. So that is the reason why this technique that we are discussing right now should be called plastid transformation. Okay, plastid transformation will be a more accurate term than chloroplast transformation to describe this process. Okay, because there are variety of plastids in the flowering plants, that is chloroplast is there, then leucoplast and chromoplast. Now plants which have transformed plastid genomes, those plants are known as transplastomic. Okay, transplastomic means those plants in which the plastid genomes are transformed. That is, for example, the chloroplast genome is genetically modified, then that particular plant will be known as transplastomic. Now, major difficulty in this chloroplast transformation to produce transplastomic plant is that you have to generate homoplasmic plants, and that is very difficult. Homoplasmic plants means that plant in which all the chloroplasts are uniformly transformed. Okay, to achieve this is very difficult because there is presence of 10 to 100 chloroplast in one cell and there are lots of cell in a plant, and the thing is that each chloroplast then contain 100 copies of the chloroplast genome, and due to this the homoplastic state to achieve homoplastic state is very difficult. Okay, these things don't allow the homoplastic state to occur. Okay, the presence of 10 to 100 chloroplast in one cell, and then each chloroplast will contain 100 copies of the chloroplast genome. Apart from this, getting a high level of protein expression even though the gene copy number is high is another problem. So that's all for this video. Thanks for watching. See you next time.